Okay, so we're going to go over, this is going to be a review for Unit 3, Biology, and it's a Unit 3 exam. So if you use this, I'm going to try and talk quickly so that you don't have a long video to watch. But you can certainly go back over it and pause, and I'm also going to list some terms a little bit later on that if you know them well and you know what's on these slides here for my PowerPoint, you'll do very, very nicely on the Unit 3 exam. So we want to know what an optical microscope is, and that's something that a microscope that uses bright light illumination and multiple lenses to actually magnify whatever you're looking at. Now, an optical, it deals with light and lenses. Well, it can either be simple or it can be compound. Probably today, if you've used a microscope, you've used a compound one, has the light source built right in. I've had a simple one that I actually got when I was in probably junior high or high school, and it didn't have a built-in light source, and it just had didn't have a lot that you could do. I think I had just some basic magnifications as far as the lenses, but I actually had to use a light, put a lamp next to my desk, and then use a little reflective mirror to get the light to come from the lamp source down to my reflective mirror and then up through the slide so that it would illuminate whatever I was looking at. So an optical microscope deals with having bright illumination, multiple lenses, and here's an example of one thing you can see. I don't know if you know what that is. An amoeba with all those little pseudopods beginning to protrude out. Remember, that's the one that looks like a blob and it's continually changing shape. Now, another way of using a microscope is to not use the bright illumination, but it's called dark field microscopy. And as the name, certainly tells, it's a technique where you have a black background, and the only thing that's really illuminated is the organism or organisms that you're trying to look at. And what's nice about this is that if you use the bright illumination, excuse me, and you want to look through a slide, usually you have to have your whatever's on the slide, whatever you want to look at, you have to have stained it so that it shows up. But when you use dark field microscopy, you don't have to stain the organisms. It's a method where just the organism shows up, but the light reflects off the organism, and it has that nice black background, so you can actually see the organism very, very well without staining it and without having bright light. Now, make sure that you know when you use a optical, or which compound is an example of it, microscope, you have different powers that you can use. Oftentimes they start with about a 4, where they magnify 4 times what the normal size is, and then maybe you have a 40 or 10 and a 40 and a 100. So there is a difference when you look through the eyepiece as to what you're going to be seeing if you look under low power like a 4 as compared to a high power, maybe like a 100 times magnification. And let's take a look at this. This is probably maybe a 4, maybe even a 10. You can see quite a few of the organisms, but you can't see them in great detail, but you can see quite a few of them. Now, if I go to a higher power, I'm actually going to be able to see each of the small things. It actually shrinks down the field that you're going to be able to see. So maybe I'll take just a little bit right here when I'm on the very, very high power. So I'm not seeing everything around it, but that one little area that I can see is greatly enhanced. It's quite a bit larger. So under high power, the amount of area you see is smaller, but the parts you can see look a lot bigger, so you can really see in detail what each of the individual parts look like. So now you usually always start with low, and then you work your way up to high power. Now, an electron microscope, this is an example of it here. There are some microorganisms that are just too small to be seen with an optical microscope, or another way of looking at it too, you want to use an electron microscope for that, or it's also even some of the things that you can see with a compound microscope. If you use an electron microscope, oh my gosh, the detail that you can see is unbelievable, like what you're seeing right here. Now, this is a flea. And look at the detail that you can see. You would never know that that's what a flea looked like, if we just use our normal sight. Probably under compound you could see, more, But under an electron microscope, look at the details. You can even see the little hairs sticking off here. Now this is pretty amazing. This here is actually a virus. There's a virus. So all these are virus particles. And so if you remember correctly, a virus particle is just like a protein coat around on the outside, and all it has on the inside is genetic material. That's it. That's all that makes up a virus. It actually has to get into a host to use all of its DNA and materials in there to actually replicate. But isn't that an interesting little thing that you can actually see that, a particle of a virus, using an electron microscope? Now this Robert Hooke was, if you really go back quite a bit, we've just talked about an electron microscope being able to see amazingly well. Well, back here, Robert Hooke was one of the first people to come up with it. He probably made this himself. There's that little eyepiece, and he actually looked at a piece of cork. And so this is what it looked like to him, very, 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 one of the very first microscopes, very primitive. But he could actually see little boxes in there, and he called those little boxes the cells of cork, because remember, all living things are made up of cells. So Robert Hooke was the guy that first came up using, developing a very primitive microscope, and actually determined that living things are made up of little things, that look like little boxes, that he called cells. Now the cell theory, here's two other, and you can tell by the pictures that these are not updated people. These are people that lived a long time ago that came up with another theory, and that theory is the cell theory that states that all living things are composed of cells. Now you might have one-celled organism, or you could have, like us, a multicellular organism, but all living things are still composed of cells because the cell is a basic unit of life. And they also came up with a theory, which is certainly true, that cells arise from pre-existing cells. And we won't talk about it now, but there's some pretty neat little, very simple experiments that they use to show that life just doesn't spontaneously arise, but that cells actually do arise from pre-existing cells. Now these two gentlemen, their names are Schleiden and Schwann, or Schwann and Schleiden. And these are the two guys that are as far as historically, are said to have come up with a cell theory. And again, you'll want to reread through here to determine or to memorize exactly what makes up the cell theory, okay? Now here, I thought this is kind of fun. I'm going to let you kind of go through some of this. We want to know what the organelles inside a cell are. And if you look closely, you'll see mitochondria, lysosome, reading on down, a nucleus, ribosomes. You may know a lot of these already. And here's where they are in here. Like here's number 1, and they want you to determine which one is that. Here's number 2. Here's number 3 in the cell. What is that? So I'd like to have you go through after you do this. Let me know, send me your answers, and I will send you the correct answers back so that you know you've done it correctly. And then I also would like you to find out, what's a plastid? And where would you find it? Would you find it in an animal or a plant cell? And your lessons are a great place to find things. Also, Google is a great place to find things. You can find some pretty neat pictures in the images through Google. Okay, a plasma membrane. Both animal and plant cells have a plasma membrane. There is something different about a plant cell. Not only does a plant cell have a membrane, but it also has a cell wall. Okay, so you can see here, and I pointed right to it. You'll notice in an animal cell they have the plasma membrane, but there's no cell wall. Now, both of these having the plasma membrane, we'd like you to know, what makes up the plasma membrane here. And so you can see there's 2 layers here, and they're kind of tail to tail is what they are. You'll notice that the head here is on the outside, and completely on the inside is the head on the other, on the inside of the cell. And together they make up they call it a bipolar phospholipid membrane. So take a look at this. It shows you what part is which, and know that the membrane, whether it be in the animal cell or in the plant cell, but remember the plant cell also has a cell wall around it. So in the plant cell, the plasma membrane is underneath the cell wall. But know what makes up both of them. Now we need to know what a hypotonic versus a hypertonic solution is. So this is kind of interesting. Now this is a red blood cell here. Okay, so what they've done here is they've put a red blood cell in an isotonic, a hypotonic, and a hypertonic solution. Now, if somebody's hyper, it means that there's a lot going on. So that's how I kind of remember hyper means that there's a lot of solute in the water there. Okay, so around the red blood cell, there's a lot of solute particles that are actually in the water. We call it hypertonic, and it could be multitudes of things that are making it. It could be sodium, all kinds of minerals, all kinds of a lot of things. That could be the solute that has dissolved in the water, but that's not what's important right now. We just want to know the difference between hypertonic and hypotonic. So with a hypertonic, you have a lot of solutes that are in that water around the cell. And hyper, remember, means a lot of activity going on, so that's the one with a lot of solute in the water. Now hypo, since there's three, but iso means the same. So really in an isotonic solution, the amount of solute in the outside there around the cell that's in the water is about the same concentration as in the cell. Iso means the same. Now hyper, meaning a lot of solute in particles in the water. Hypo would be the opposite. So it would be very little solute particles that are dissolved in that water around the cell. So you can see that when we talk about a hypertonic solution, we're talking about a comparison here, that there's a lot of particles here in the water as compared to the amount of particles in the cell. This one, the hypotonic, very few solute particles in the water as compared to what's in the cell. Isotonic, the amount of particles in the water around the cell is about the same as the concentration of particles in the cell. Okay, so I was going to skip on by it, but I believe that it would be good for us to know where the water goes with, if we have these particular situations. So let's kind of look at it here. When you have an isotonic solution, usually water will go from an area of high concentration into an area where there's not so much water. That's just how water works. But remember, in here, we have water in the cell too, and we have a certain number of particles in the cell, and we have water out here with a certain number of particles in the cell. If the particle concentration is about the same inside and out, then the amount of water inside and out is about the same here. Inside and out too. So nothing really happens. Not a lot, or some water can go in, some water goes out, but it stays pretty much constant as far as the amount of water outside as compared to the amount of water inside. Now here, take a look at the hypotonic solution. The hypotonic solution had very few solute particles on the outside, but there's quite a bit more solute in here. So where is there more water? There's more water on the outside because there's very few particles. So that makes me think, oh, there's more water on the outside. So what's going to happen? If there's more water on the outside, it will come into the cell, and it actually can swell up that cell, and if enough comes in, it can actually make the cell burst. So that's what happens if you put a cell in a hypotonic solution. Let's compare that to a hypertonic solution. Remember here, we have a lot of particles in that water around the cell as compared to what's in the cell here. And so there's very little water. There's the particles are taking up so much room that there's very little water. That's how I think of it, and that'll help you remember it. So if there's very little water on the outside, because it's so many solute particles, where is the water going to go? Well, remember, the concentration of the solute is in relationship to what's on the inside here. And we have tons of solute on the outside as compared to what's in the cell here. So since there's tons of solute on the outside, there's very little water. So in this case, the water from inside the cell would actually move out to try and give that outside area more water. So what can happen with a cell that you put into a hypertonic solution is that the cell can actually shrink down. Okay, so try and review in your mind what a hypotonic versus hypertonic as compared then to an isotonic solution is, and review again, if you put a cell in each one of those, what will happen Will a cell remain the same? Will water go into a cell and maybe even cause it to burst? Or would a cell shrink because water is actually leaving the cell and going into that area around the outside that has so many solute particles but hardly any water? Okay, so let's continue. We need to know that and which you probably know, the nucleus controls cell activity. Here's the nucleus here, here's the nucleus here with a little nucleolus in it. And that's where, kind of like the brain of the cell, and it also has all the genetic material in it, so anything that's going to take place, from building of proteins to replication to anything, all of the instructions will be found within the nucleus of a plant cell and an animal cell. And you'll want to know that the cytoplasm, which is just all this it's kind of like the base that all the stuff is floating in here too. The cytoplasm and the nucleus together are called protoplasm. Okay, now here's where I listed some terms for you to know, and I want you to go through your lessons and look up what these mean. If you do so, you'll see them on your test and you'll be able to do the multiple choice or fill in that deal with these terms. There are also some matching. What I've done here to make it a little easier for you, I've put the words here: organism, nucleus, nucleolus, and I've told you what they're going to be matched with. So if you write those down and figure them out, you'll have a good head start on the test, because you're going to be seeing some matching with these terms and these definitions on your test. There's certainly, and I'm sure you know that there are specialized, very specialized cells, like we were looking at earlier. Remember the red blood cell that we were looking at in an isotonic, hypertonic, and a hypotonic solution? These are red blood cells, what they look like. This one here, these are kind of misshaped here. I don't know if you've ever heard of sickle cell anemia, which can be found in African Americans, in black Americans, or black people in Africa or wherever they are. It seems to be something that's just found in black people. And the red blood cell can actually get sickle shaped here. And it actually is called sickle cell disease, and it can be very painful, because you can imagine that if the cells are not the proper, round shape here, and they go through the veins and arteries, that these sickle cell shaped red blood cells can start plugging up things and can cause a lot of organ damage to the people that have it, and can cause a lot of pain. And there are certainly different degrees as to how severe or how non-severe a person may have it. But that's a good example as you're looking at these red blood cells, which are certainly specialized cells. That's what carries oxygen. So you can imagine that it also, when they're sickle shaped like that, they don't carry oxygen as well as they should. So another problem with sickle cell anemia. So here's a picture of all the red blood cells. Bone cells, you can see they certainly look different than red blood cells. So specialized cells, they usually perform a specific function. And you see that in a multicellular organism. You're not going to see that in a one-celled organism. So like we as humans have many, many, many specialized cells. Level of organization in biology, you need to know that, you certainly start out with, remember, all things are made up of cells. There it is. Cells come together to make tissues, whether it be skin or even though skin is considered an organ, it can be just different tissues that make up the heart. If you put tissues together, you could get the complete organ, like the heart itself is made up of many different tissues, and those tissues are made up of many different cells. If you put a lot of organs together, you get a system. Let's see, we're talking about the heart, so that would be like the cardiovascular system. And then the cardiovascular, all the different systems, including that one we talked about, the cardiovascular system makes up the organism. So here they're talking about, here, it looks like the kidney, so the urinary tract, the urinary system. Okay, so you can see that we have cells making up tissues that actually make up this is a kidney here, and the kidney comes over here as the kidneys, and there's a ureters bringing, the urine is formed in the kidneys there, and brings the urine down through the ureters into the bladder, where we hold it until we pee. And that is just one of the systems within an organism. There are many, whether it be the cardiovascular or the the urinary system here. So just kind of memorize these, the level of organization Starts with cells to tissues, organs would be like the third, systems would be the fourth, and the final organism would be the top. Know the difference between prokaryotes and eukaryotes. Eu means true. So that means we have in eukaryotes, you have a true nucleus. And if you look here, here's a eukaryote, and if you compare it to a prokaryote, and a bacteria is a good example of a prokaryote. I see a nucleus in here. I don't see one down here. There's genetic material in here. It would be there would be DNA on the chromosomes here. Down here in a prokaryote, they just have one single strand that carries all the genetic information in a prokaryote. So they want you to know, the main differences between a eukaryote and prokaryote. I put them here. You don't need to memorize everything. Just know that. What else do you see different here? I see the nucleus. I don't see it here. It looks like there's some organelles here. Looks like there's a mitochondria. There's an endoplasmic reticulum. I don't see that down here. So you actually have membrane bound organelles up here in the eukaryotes, but you don't down here in the prokaryotes like a bacteria. So know that bacteria and archaea are the two things that you will find in the prokaryotes. They used to actually put the archaea, they actually considered it a bacteria. And you don't have to go into the specifics, but there are some differences between the bacteria and archaea. But the main thing you need to know is that they don't have that nucleus, they don't have membrane-bound organelles, and so they fit down here in the prokaryotes. Negative feedback versus positive feedback. This is kind of interesting. Let's see if I have everything I need here for you. Probably we're more familiar with negative feedback. So negative feedback sounds to me, what does it sound like to you? If you have a positive feedback, negative feedback, it almost sounds like negative feedback is not going to be going the direction maybe that we're going. It's going to go the opposite. Positive feedback almost sounds to me like maybe I'm doing something and it's going to keep me going in that direction. And indeed, that is exactly where it goes. And what I've given you here are a couple examples. Say that you have, here's negative feedback. Say you have, maybe you have a fever, or you're out outside and it's 106 here in Boise, and so you're getting pretty hot, and the body temperature raises too a bit. What we have, the reason that our body temperature doesn't get too hot outside in that temperature is because we have a negative feedback system that works so well for us. So what's going to happen is that we're going to sweat. We're going to start sweating more. And if the more the body temperature rises, the more we sweat. And I don't know if you realize it, but when the sweat evaporates off of your skin, that's what cools your body down and drops your body temperature. So if your body temperature starts to raise, so it's going in one direction, we want to go the opposite direction. We want to go, negatively. So our body, one of the feedback systems, what our body does is we sweat more, and that causes, our body to be nice and wet on the outside. It then evaporates, which then lowers our body temperature. So you can see that we're going the opposite direction from what it began. The body temperature rose. We did something with our negative feedback system to actually lower the body temperature. We went the opposite way. Now another example would be this one here. And before we go through that, I thought a pretty good example would be like during childbirth. We have, someone is expecting a child, and you have the cervix, that's going to have to open, the cervix is right at the top of the vagina. It's going to have to open so that the baby can come out. So what happens is after 9 months, that cervix starts to expand a little bit. And what the body does then, when the cervix starts to expand, certain receptors sense that the stretching is going on, and it actually increases then the contractions, and the contractions then make the cervix expand even more. So you can see we're going in the same direction here. We started to have a little bit of expansion of the cervix. It went through the process, and whatever happened there actually made the cervix expanding even more. So that is an example of a positive feedback. We went in a certain direction, and the feedback process actually kept us going even further in that same direction. Let's look at the example here, another example of positive feedback. I put money in the bank, and the more money I put in, what happens? The more interest I earn. And the more interest I earn, what does it do? Goes back in, that money goes back in and makes my balance grow, and then I earn more interest. And that interest now is included in the amount of money I have in the bank, and I get more interest that I earn on it because I have more and more money all the time. So that would be a positive feedback too. We're going in a certain direction, and whatever happens, whatever the process is, keeps us going further and further in the same direction. So I think if you really think about the terms, they kind of give you a hint as to which direction you're going to go. Between the negative, nope, it wants to take us not the direction I'm going, but backwards. And not that it's negative, because as you can see, negative feedback is a good thing. It's just that there are two different types of feedback, both being good. And then the positive feedback, whatever direction we're going in, we keep going in that direction. So I hope this helps. Go ahead and send me or let me know when you're finished with this. Send me the answers, and I will let you know if you're correct on that one slide. Go and look those other things up that I had mentioned and review the slides I've given here, and I think you'll do very, very nicely on the Unit 3 exam. So let me know how things are going and if I can help you in any way, and thanks for coming. Bye.